Welcome back and Happy New Year. It's 2026.
It's 2026. We haven't been here for a while.
No,
No.
we were freezing.
It's freezing cold in our cottage where we record these, but we're excited to be back because we had an important interview.
We did. We had the pleasure of speaking with Dr. Lou Wing, who taught us all things epigenetics and theology in his one book, Genes of Eden. We had the pleasure of reading it and also had our work cut out for us in learning the language and the terminology, the science and the theology.
It's awesome. Great interview. You're going to get a lot out of it. And you can't help but not love Lou. He's so
awesome.
Fun. Got the greatest personality. And you get this, you just have this appreciation that God put Lou in the science space, and he's a pastor, and he's just excited about how God made us. And so just by way of background, he is a biochemist, he's a toxicologist, and he spent more than 25 years in the biopharmaceutical industry. He's a consultant and a pastor. And as Crystal said, he's written this new book. So that's what we talked with him about. Please enjoy the episode. Dr. Lou Wing, although I know you as Lou, thank you so much for joining us. And right off the top, Lou, I got to ask you this question. We're going to talk about the science and the book, but I want you to explain to me the mice who smelled something horrible, and then 2 generations later, their offspring had this negative reaction to the smell. I think this in your book is the Dias and Reiser study. Let's open up with that. Exactly. Tell me that crazy story. What's going on?
Okay, so this is one of the things that can happen with generational trauma or generational, any kind of incident, environmental incident that can find itself imprinted within the genome. And this is kind of like the whole aspect of how this concept of original sin, we'll get to that, but just the idea of that study is evidence that a change in the DNA function can have an imprint, and it can be seen not only one generation, but now we're seeing up to 3 to 4 generations just from experimental procedures alone. All the gene editing studies I've been doing, I've been a vice president for cell and gene therapy for many years, and with Parexel, which is a East Coast company. It's an international consulting firm. And one of the greatest challenges with gene editing, and just like we're discussing here, imprints within genomes, genes can be shut off and they can be environmentally altered without changing the sequence of DNA at all. So mutations in DNA are extremely rare, and the rarity is seen because of the lack of structural change, but there's a very dynamic process that happens to the function of a gene. So just as a basic idea about, we have the genotype, and remember in the phenotype, well, there's something that's happening in that distance between the structure of DNA and what the function looks like. And that's really where my world is the function. What is this gene doing and how is it adapting? And the mice study is a classic example of how that happened. There's also the agouti hamsters and so on that have the skin color based on trauma. These kind of variations that we're seeing is one of the major thrusts of why and how. What is this concept of sanctification? And that's kind of what we're going to go to. But just know that exact study plays right into the whole subject matter that we're dealing with right now.
Okay, before we talk about science, let's talk a little bit about Lou. And so you've written a book called Genes of Eden.
Yeah.
And we've got it here. It's all marked up. And
Yeah, good.
is
Wait,
so walk me through. So you're a scientist. You're also a pastor.
yeah,
You're also like a
yeah,
professor,
yeah.
speaker. You're like this. You
Mhm.
got all these different things that you're doing. Um,
Uh-huh.
is
Yeah.
this book for you? How'd you get to it? Like, is this in the last 5 years? Or is this something that you've been working on and it finally came out in a book after 15 years of work? Like, give me a little bit of chronology on Lou.
Right, where your chronology is dead on. The only thing is 10 years. And so
Okay.
I go back to about 10 years ago when I'm in this arena of, like I say, cell and gene therapies. I come out of the Bible college with you with, took a little, Selah of my life to study and look at some of the Old Testament's world of, Leviticus, for example. And as teaching through that, got a better handle on how can there be an interface of science and the tools that we have to be able to look at scripture and have that understanding of, what happens like with that peace offering concept and, how these ideas sort of work their way into how we understand the Word of God. So I mused over that. And then by coming into this arena of epigenetics, I went, oh my gosh, this is why this extraordinary imagery of I don't have to arrive as a scientist to the evolution creation debates anymore. Those were the most fruitless times because nobody wins. And it wasn't even debates because there was all about the heart factor. And so I thought, well, what would happen if I saw science as a tool, as we know it now? And understanding the sciences that we know now, I don't have to explain origins. I don't have to explain where we came from. I don't have to be limited to Genesis 1 through 3. I can actually see science as an unveiling of how God is working in now. Like, how is this science working now? Which is why I went into pharmaceutical sciences in the first place, because I realized our science is giving us tools on how we can manipulate some way a mechanism that's occurring in our bodies, whether it be cancer, autism, or any sort of disorder, if you will. And if we can use these small molecules and they can manipulate it, is there something that's happening in us? And that's what really hit me is like, what is happening in us, even with this concept of, I had to mention sanctification a while ago. Like, how is that affecting my body? So actually looking at First Thessalonians 5:23, just I guess that was a 10 years ago moment that I had thought, the God of peace sanctify you completely, mind, body, soul, and spirit. It says that point blank. Where is the body coming from? So I thought, I need to get more depth in theology. And so I went back to Aberdeen University where I did my PhD in biochemistry. And the theology department there is extremely intimidating and highbrow and whatever. But I had this professor there that was extraordinary. He ran the disability theology group, and he has a Down syndrome child. And he saw my thesis idea, and he said, "Oh my gosh, you've got to do your graduate degree here in this, and you have to explore it." And the best theologian to look under is Augustine as a natural philosopher who didn't have the tainted, evolution mindset. He simply saw science as it is. And all of his theology that surrounded that, his concern wasn't origins. His concern was how do we explain how these and his focus was original sin, how is that integrated in us and how is it propagated in us? He uses the word propagated, and I went, oh my gosh, that's a genetics term. So I realized, okay. And then he even goes on to say, there's a quote in the book about the fact that he says, who will ever explain how this can possibly occur from the sin of Adam to be in all humanity, according to Romans 5:12. So when I really studied that verse and looked at the fact that human genome tainted with sin, how could that occur? The same concept with how does the soul exist because it is tainted with sin and God isn't sin. God knows no sin. So this early stage of understanding how the soul exists in us, how the soul is propagated, how the sin in the soul is propagated, coming from Tertullian to origin and the space that happened within this concept of traducianism, which is the understanding of the soul, material soul, passed through generations. It was dismissed for millennia because there was no way we could ever see the sort of, metaphysical aspect of a materiality of the soul. There was never any concept. So we immediately, from the Catholic Church on, kind of kept a look at that, but was completely dismissed by most theologians. And they looked at the creation factor of, that are just like we're created, so is our soul, and they all come in one package. But interestingly, as the story goes on, is that my passion was understanding if I can find out the way we can see how the soul could be propagated, then we can kind of make an inference of how sin is propagated.
Hmm.
And in fact, if it is in us, the most logical place this soul tainted with sin would be by an epigenetic phenomena. Why? Because epigenetics is a changeable. Yet, and as we talked about the mice study, it also has a permanency that can also be generational, but also changeable. So there's this dynamic that happens in epigenetics that gives this credence to the fact that we are not only positionally sanctified, but we're also progressively sanctified because there's this changeability in our genome. So that the concept of being holy is a reality scientifically. So I thought, okay, wow. So we can take this down. How can we actually see this in us, like who we are? Well, the best evidence, and really the best evidence, is from Augustine and how he explains in his confessions this evidence of sin, and it exactly mirrors addictive behavior. So when I looked at addiction and you look at it against this sort of outflow of original sin, it shows us that we have all this potential within us. And so I looked at the fact of the matter is we don't, I can't say that I've got, addictive tendencies because my mom and dad were. It's because you're making sort of like a structural concept when in fact I believe, and this is what the science is showing us, is that it is a changeable yet imprinted condition that's in all humanity. We all have the potential for this behavior, and yet it can be obliterated based on how changes in our epigenetics and our environment has an impact on our genes.
Hmm.
So I hope that explains some of
No,
the.
it's good. I have more questions. Do you want to take a question?
I have so many questions. I'm going to
Okay.
start with with one that I'm super curious about. So, I feel like anytime you try to combine scientists and people of strong religious beliefs in one place, it's a recipe
Yeah.
for a lot of fun, and sometimes messy
Yeah.
fun. And I think you're a brave
Yeah.
man. So I guess that's just really my comment, not so much of a question. You seem very brave,
Yeah.
willing to do that. And
Uh,
along that same line, as I was reading through the book, I noticed that Augustine's relatively well accepted. But there was at
mhm.
least one point where you referenced some theologians who had some more creative and outside of the box ideas. I think it was Origen was one.
Mhm.
What
Yeah.
what prompted you? What made you brave enough to consider those thoughts, those perspectives, and how did that shape the outcome of the book and then the reaction that you've had in speaking with other
Excellent
people?
question. Exactly. Okay. So, um, looking at the chronology is really one of the most important factors and sort of tracing how because I didn't just come up with this. This goes up the 120 AD is when a lot of these theologians, these early church fathers, were wrestling with this exact issue. And it was from Tertullian, then it came to who was sold Traducianism. Traducium simply means materiality of the soul. It's the actual word means to pass through. So this whole idea is that the soul is the material passes through the
Mm,
parents, and that's where the soul originates, and it stays within us, and then it's again passed on materially. There was no explanation for it. It was simply this Tertullian. Origen had a more mystical perspective in terms of, yes, that possibility is there, but it was more or less this concept of creation and pre-existence of the soul. So it was this more ethereal. Origen was a very sort of out of the box, theologian, and a lot of his detailed, ideas in terms of how he would explain any types of theology. There's been a lot of controversy in light of, just his dogma in general. Although his first principles, he does give some explanation of how a soul could be sort of existing and it could be sort of embedded within us, but there wasn't any sort of concept of that. Well, along comes Augustine in 300 AD when his idea was, yes, that could be possible, although I'm not going to commit to it. So he was half Traducian, half pre-existent, and very little bit on the creation. Then along comes Thomas Aquinas about 1,000 years later, and Thomas Aquinas had this idea if there's a subsistence of the soul. So it's a sort of material and non-material. So again, he never could really commit to either one, but the Catholic Church embraced Aquinas's work, and that's where the Catholic Church sort of builds itself even today around Aquinas's concept of the subsistence of the soul. They're not committed to it, yet they're open to it as being a creation, but they still stay away from the idea that it's a sin, could not be given by God. Well, along comes then we get to Luther, and then we get along to the Reformation, and then ultimately, there was a soul concept that was all about creation, and that's where the soul is, and that's all it could be. So all the way up now, and when we're talking, it was not even discussed really until we come up to the present day. And interestingly, Norm Geisler is a full-on traducianist. And I went, oh my gosh, okay, so he really held as like a number one apologetics guy. I went, could he possibly think this? But he said, interestingly, in his last writings, he said, someday there's going to be some more explanation about how this traducianism works, and it could possibly, he even gave a foreshadowing of a genetics function. He didn't say epigenetics, but he said something along the genetics function has got to have something in that arena. Well, my concept is that yes, but also it would be called a neo-traducianism, meaning a new form, just like neo-Darwinianism is all about DNA, which is a whole subject matter that's happening right now, a very current topic. However, neo-traducianism is an epigenetic phenomena related to a traducianistic perspective, not pre-existent, not created. It's yes, God forms us, God makes us, He gives us, but He also, within that concept of free will, there was this our idea about a soul and how sin is something that we're not only embedded with, but it is evident that it doesn't take much for us to figure out that we have sin from our very onset. As a matter of fact, Augustine nails it by saying we are born moving away from God. And that whole idea about that moving away is all about the distorted desires and this on where we have within us this moving, oh, like literally falling away. So I see it from that idea, that sort of the string that you're talking about, like how do other people buy into this? I've had discussions with, yes, theologians on this, as well as scientists. And interestingly, oh my gosh, the more high-powered scientists, if you will, are very open to this concept. As a matter of fact, I'm working with a lab over here at UCSD, and they're doing gene therapy with a compound I talk about in the book called the brain-derived neurotropic factor. It's involved with learning and memory, and also it's very involved with Alzheimer's. And so they're trying to do this, gene therapy to sort of get more BDNF made in certain regions of the brain to stop the process of Alzheimer's. And it's a really interesting early studies show that it's actually working. Well, this BDNF is a paradigm, literally, of how we can see how this is a very epigenetically dynamic molecule in terms of our learning and memory and environment and how it's affected by, and I'm just saying, by it's tainted by this concept of a sin nature. And
Mm.
so I talk more on that in the book in light of, decision making and learning mechanisms, and always with this concept of, it's fascinating is that, this issue of doubt from the very onset of humanity. We always had this potential to sin. And so the trigger that we see in Genesis 3 is the trigger of doubt and doubting of God's word, and the idea of, the lack of acceptability of an absolute nature of God's attributes. So I see that this book playing into a lot of that early stage of doubt, and then understanding that this sanctification process is addressing that and giving us a greater perspective on the wholeness that God wants to give us, to develop in us through this biochemical mechanism.
So, Lou,
So,
let me see if I got this right. So, you had a degree in biochemistry. Were you a
yeah,
doctor of
yeah,
biochemistry, or did you?
yes, I have a PhD in biochemistry. Yep.
Yeah, but
And
then you went back to school.
but yeah, I went back to the same university and did my master's in theology
Okay.
with Brian Brock. Yeah.
Okay.
And yeah, yeah,
Wow.
yeah.
And you're
And so
connecting as you're
the
going through it.
two
You're
of
like
them.
connecting all the dots. Okay, let's talk a little bit about
Yeah,
science for a minute. Is that okay?
Of course.
yeah,
Okay,
Yeah.
I got a
yeah.
prop
Oh, good for
because
you.
yeah,
I'm
because people
impressed.
people are not going to track with this. I mean, it took me a minute to like catch up on the science. Okay, so
No problem. Yep,
we have a genome right here.
yep,
Okay, and on a
yep. That
genome,
that is a segment of the genome. Yes.
this is a segment, and so people are familiar with the double helix, right? And we
Right.
have
Yep.
on here nucleotides. Is that right?
Good. Yep.
And there are only four, right?
Yep.
There is AT and a CG, and C has to go with
Good.
G, and A has to go with T, right?
Excellent.
Right.
So far, you're the MCAT. Go on.
So,
Yeah.
so this is what we call DNA, but
Yeah.
this is not epigenetics. Epigenetics is
Okay.
describing how this expresses itself, how it functions. I think that's the word you used. How this
Mhm.
functions in the body, how it manifests itself in the body. And in this book, you're specifically focused on methylation, which is the
Mhm.
C, right? I don't know if people can see this on here, but
That's
that's
awesome. Yes. Good. Perfect. Yes.
is that right? Are we got do we have
Yes.
the science
You're
right?
nailing it. Yes.
What
I'm
else do you
loving the model and all that.
Oh
Now,
good,
wait a
thank you.
minute. Now, you had a red dot. You had a little red dot on the orange one. Is that the C? The
Yes.
cytosine?
No, we the
Okay. So you're showing the
C is a
methylation.
orange. Yeah, C is orange.
Hmm.
Yep.
Yeah.
Yep. And that little red dot is your methyl group.
Okay.
Right? See the little red dot there? So that would be your methylation.
Okay.
Because the C is the only one that gets that methylation means CH3. So when it sticks to that C,
Yeah.
it will prevent it will prevent other molecules from attaching to it. When that methyl group is gone, it's like an on and off switch. The gene can
Um,
be expressed.
Okay.
So you have those methyl groups on C, and okay, just a little more biology here. You have about 25,000, let's say, genes in your genome. The genome is all your DNA, 6.4 billion base pairs, just like you showed me, A, T, G, and C. There's 6.4 billion.
that's a lot.
Only 1% of that 6.4 billion is genes. The rest of it had been considered junk DNA, when in fact, all of the other 99% of the DNA is not junk. It's all involved in how the genes are expressed. It's part of the epigenetics as well as DNA methylation. So we have, interestingly, okay, DNA makes RNA. RNA is a simple segment that is one strand and it's mobile and it's referred to as a messenger RNA. A messenger RNA leaves the nucleus, goes into the cytoplasm of the cell, and it goes into a manufacturing plant and makes a protein. And that's what makes up our body. That's the basics in molecular biology.
Hmm.
But interestingly, all of those, okay, those are messenger RNAs carrying a message. That is the message of the protein. There's a lot of also, okay, here's the freaky part, of non-coding RNA. In other words, RNAs can be made around from a lot of that junk DNA, and those non-coding RNAs are actually part of shutting a gene off. So it will sit on top of DNA and not have it expressed.
Hmm.
Okay, so example, you and I, Josh, we have two X's just like Crystal does. And yet, interestingly, our other X chromosome is silenced. Okay, there's a number of genes in our bodies. Okay, we have 256 different tissues that make up our bodies.
Hmm.
Okay, and of those 256 different tissues, a neuron is different than a kidney cell or a liver cell, but they all have the exact same sequence of DNA, meaning that epigenetics tuning, if you will, with the RNAs, silencing RNAs, as well as the methyl groups, those genes are turned off, the ones that aren't part of the neuron that are in the kidney cell. That's the freakiest part. Every single cell of your body has the exact same sequence. It's just a matter of how it's tuned.
Mm.
Okay, so here's the freakiest part is that behavioral genes are probably the most dynamic besides the immune system, epigenetics. And why behavior? Because the environment that we live in, our behavior is the most
Mm.
evident by that change. Because other genes like growth genes are also highly dynamic in terms of epigenetics. That's where we get cancer. Okay. So a lot of different types of genes are more malleable to environment. Like PTSD is a classic example. Autism is epigenetics. These are things that have a impact on based on their epigenetics. Are you following me?
Yeah.
Kinda.
Yeah.
Mhm.
I'm not going to give you a quiz. You don't have to be responsible for this, but just act interested. That's all I need. Okay. So once you kind of get this idea going, though, you realize, okay, the behavioral genes that have these epigenetic changes can be influenced by an environmental factor. It could be a traumatic experience, dietary changes, hunger, all the stuff that I mentioned in the book as far as how these changes can occur. And they can be severe enough to be generational, such as the Holocaust and
Hmm.
the survivors of the Holocaust and how their story was imprinted into their genome and has been recognized 4 generations later in dreams of great 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 grandchildren of identical incidents.
Wow.
That
Whoa,
that
that's
alone,
trippy.
Rachel Yehuda's it's documented. And Rachel Yehuda's got I've listed her as a reference and all that. Fascinating work that gave more freedom for me to sense, oh my gosh, so what would happen from the original sin then? Is it an imprinted phenomena that's in our genome and is altered periodically following salvation, sanctification? All these concepts that we keep sort of like in our Bibles are actually a somatic functioning aspect of who we are as believers. And the closer we get in walking with Jesus, the transformation that's happening is not only spiritual, but it's physical.
Hmm.
And that's where these genes of Eden is so potent because it gives us an understanding that we are not our own. God bought us with a price. He's transforming us. He transforms our minds, our bodies, our souls. And in the process, there's certain things in our lives that become altered and or are changing based on the circumstances of whether it be in the word, whether prayer is a classic example, things that are happening in this concept of we say sanctification. That's a
Um,
lot of biology.
that's a lot.
You
I have some
opened
more.
up.
I just don't want to cut off Crystal because she's always got the better questions than me.
No, that
No,
is
that was
not
a really
true.
good one. You guys are doing great.
Not true. Go ahead.
Let's stay in the realm of science. Um,
Go ahead.
Yeah.
the because I want to do science, then I want to do I want to jump over to theology. Um,
Okay.
so you're looking at this realm of epigenetics, and you're seeing
Hmm.
these studies that are being done. I feel
Hmm.
like we've mentioned it. We talked about it with the mice, but one of the the key tenets in this whole hypothesizing about like maybe did God make us this way is this idea of inherited traits. And so
Uh-huh.
could we just punctuate that fact again and just explain the difference between an inherited gene from our parents and an inherited epigenome? And
Hmm.
because I think that
Oh, that's
we always talk about like you hear people say like, oh, I got this gene of addiction, or I got this, I got the
yeah.
gay gene, and people go back to like genes, which we know we got from our parents, but it seems like
Hmm.
the breakthrough study and a lot of what like your material hinges on is this idea of also inheriting an epigenome, which is not so locked in, right? The epigenome can change based on trauma, but also the person, like I can live a healthy lifestyle and change environmentally my own epigenome. Am I getting that right?
I got excited. Can I
Yeah,
interrupt for
absolutely.
just a second?
Yes.
And Lou, tell
Yeah.
me if I got it right. So I kept writing notes. It was actually kind of funny in the book. I wish you could see it up close because there are like a lot of pages and underlined and notes and questions. And one of them, and we'll see if I
Did you
remember right.
rewrite it? I mean, sorry. Go
No,
ahead. Yeah.
no, I should send you a copy though. You can see.
Okay,
So
good. Yeah.
it'd be like a whole conversation. Um,
Mhm.
but so
Yeah.
what I learned is that epigenetics is changeable and malleable, and that's what gives us hope ultimately. But that
Hmm.
genes are not. So genes are permanent, but epigenomes, is that the right word? Are
Yeah.
changeable. Is that right?
That's excellent, excellent, excellent. Okay. Yes, on both of your accounts. You guys got it. Now
Yay.
here's the big issue.
Yeah.
Okay. Okay. You're absolutely right. We inherit traits from mom and dad. That's exactly right. And this is where we get this phenotype of what we're looking at right now. And okay, there are interestingly 100 to 300 genes specifically in the dad. In our sperm, we have the greatest potential of imprinted genes that can be generational. Interestingly, the eggs of a woman are clean of epigenetic marks. So all of the ova of a woman have no, and this for except for there's only a few that we're finding out now that have epigenetic imprinting. It's the sperm that has the imprinting. So when the sperm and the egg come together, we have the mix and match of all the different genes of mom and dad, and the imprinted genes stay the same generationally. Okay, and they did the study with the IG, the insulin growth factor gene. It's one of the ones that they trace the most because it's the most dynamic and it's the most related to certain other disorders, sort of in the pharmaceutical world, whatever. But that's where they're showing the imprint number of genes that are imprinted that stay with the new egg. Okay,
Mm-hmm.
so that being said, then that's where the traumatic experience that happened with the guys comes, and that those marks are then in the new child. Okay. Which makes this as a sidebar and a footnote really interesting is that if the egg is pure and has no epigenetic marks, it would be a perfect scenario for understanding how Jesus was the unimprinted
Hmm.
Holy Spirit fertilizes, and then you have the perfect human. I'm just
It just
saying,
so
but
happens. That would
that's
be
kind of,
convenient.
it just so happens that that kind of plays into that concept, though. Really interesting. You sort of have an answer for something
Does
like that, right? Is that
does that
it's
mean
a
I can
biological principle.
blame? Does that mean like everything that the kid does wrong is because of the father? That's what I'm hearing. That's
What I would, regardless if that's true or not, I'd run with it. No, you got this. But I would, it's really interesting. Why would the sperm only have, and that's kind of one of those enigmas that we're still kind of working it through. But all that to say is the epigenome doesn't really form until the zygote, that's the fertilized egg, forms. And that's why it's so important for a woman to take the prenatal vitamins, the folates, because folates contain methyl groups, and those methyl groups are all necessary to punctuate all the genes with these epigenetic changes. So that vitamin is essential.
Hmm.
In other words, dietary changes are very important for having epigenetic changes facilitating that process. And so this is why that example I gave of the Dutch hunger, the famine, and so on, and how the progeny were all of different heights and so on, and very small. And that's so like the outcome had a really interesting sort of phenotypic change, like structural changes that were from an epigenetic phenomena. So we have to keep that in mind as well. It could also have phenotypic changes, which is really bizarre. It's not a
Hmm.
mutation, but it's a change in function. We got to remember that any mutations that happen, which is why this whole concept of evolution dies on the vine when we talk about, all you need is a bunch of mutations in order to get this, changes in the phylogenetic tree and so on. When in fact, mutations are always harmful. There's never a beneficial mutation, and they're rare. And your body has four different mechanisms of repairing a gene if there is a mistake in the replication process. So, because of the rarity, it really throws a wrench into the concept of how evolution could have actually started. A recent book that just came out by a guy named Vox Day. It's called Probability Zero. It's number one right now on the New York Times bestseller list. It's a fascinating read, and it gives all of the reasons why evolution can't occur by natural selection because of the mutation frequency is zero
Huh.
with the generations necessary for us to generate humans
Wow, that's
or any
amazing.
others.
Huh.
Hmm.
So there is. I think I listened to your interview with David Gutsick, and you said that in this book you're really focused on methylation. There's also this
Yeah,
arena of histones that are
yeah,
wrapped. It's a part of like the wrapped nucleotide, and you want to
yeah,
focus on that in another work. Is it worth mentioning
yeah.
the histones just in passing?
Just in passing, and if you just want to add another flavor of confusion, that I'm so okay with that. But it's fascinating though, because that instead of a methyl group, they're called an acetyl group. That means that there's two methyl groups together, and they can have an impact on not only how a gene can function, but they're very involved with disease development and immune function. And so a lot of the things, again, that we don't see on the outside are metabolic processes and immunological properties that occur from epigenetic changes, i.e., a traumatic experience could be evidenced with methylation in behavioral genes, but also can alter immune function, and it can make one susceptible to more autoimmune disorders, lupus, and so on, that the fibromyalgia, a lot of these kind of disorders that we have in our, like, why is there such a preponderance of the cancer involved with the pancreas, pancreatic cancer? It seems to be one of the fastest growing. There's a reason in terms of epigenetic changes, in terms of how our culture, whether it be a dietary factor or a traumatic factor, cultures are influenced. This is why there's a literally a widespread cultural trauma that, just like what happened with Nazi Germany, same thing, the cultural shame that can have an effect on disorders of the immune system. That's the subsequent book I'm talking about. But the next one I'm talking about really is about our identity and who we are in Christ and so on. And what is that? How is that happening from a biological perspective in choices we make and who we are and direction and there's a whole list of stuff that we can talk about on that. But I can just say that this is like the setting the groundwork for understanding a paradigm, which is why I made it short enough to get the basic science on in terms of the methylation process. That in and of itself is complicated enough, but
Yeah, no
because
kidding.
there's right, but just recognizing that, like I say, the histones is a fascinating aspect of it, and that's really related to other types of diseases and the non-coding RNA. So there's three epigenetic phenomena that we know about: non-coding RNAs,
Hmm.
DNA methylation, and histone acetylation. Those are the three.
That's helpful. Yeah,
Mhm.
that's awesome. Um, Last
Yeah,
question in kind of the science arena, and then um,
yeah,
we'll jump over to some theology and just application of it. I was listening to a
yeah.
podcast last week with a scientist, and he was saying that there's this sense of there's been an overinvestment. In his opinion, he was criticizing the overinvestment in genes and an underinvestment in environment and this arena that
Mm.
epi, epigenomics deals with.
Yeah.
That the
Uh-huh.
funding has kind of gone towards genes and not so much a focus on environment. What do you think of that idea and that criticism?
I completely agree with that. And yes, because it's a much more understandable process to look at structural changes in DNA. This is why gene editing, which is like a number one structural change, if we can edit out, or for example, like a classic gene editing is sickle cell anemia. That's just one base pair. That's all it is. And if you could switch that or alter that or turn it off, you win. And they've done that. And they've edited the genome and taken that out. So families, you can turn it generational changes, are going to have a no sickle cell anemia. The problem is that by doing that, you also interestingly turn on a higher susceptibility to malaria. So you have
Oh,
this interesting dynamic of you don't just change a gene, you also have the consequences of altering the function of other genes. Bystander bystander genes are affected by the change of one gene. There's most phenotypes involve multiple genes. So that's why we have to remember you don't just change one gene without being worried or concerned about other genes. One of the most challenging things about these gene therapies is off-target editing,
mm.
meaning that you could affect other genes and not do what your intention was. And that's where I come in because the toxicology looks at primarily gene and off-target gene editing. So we have to look and see around the genome what else is happening to know that this outcome isn't just a simple fix on one gene. We want so bad to reduce this down to just this one chain. We just go in, snip it out, put the other one in, and it's just like changing a brick. That's not the case. And the funding is really limited on epigenetics because of the complexity. There are many factors involved in an epigenetic phenomenon. But what we know is, from what we're understanding now, is that when we can consolidate it down to let's just deal with the behavioral factor or just with the immune function and so on, you can look and we're getting a better handle on looking at a genome-wide methylome evaluation. So you can actually, instead of doing the sequence of the DNA, you can actually look at the methylation pattern. We look at it's called the methylscape. Every human has a methylscape. That's what they're called. It's like we all have that change, and that methylscape can change just like the sand of the sea does on a day with the different types of weather. You get a storm in your life, and you can see methylations in its particular genes that can change a lot of your behavior. With you, with my wife, my family, we moved and lived in Aberdeen, Scotland from San Diego. Talk about trauma. Okay, so we go to the middle of the North Pole and we're going to be living in a house in the 1700s. You honestly
Oh,
think that
wow.
there's not going to be an alteration in our genome? It's significant. And that level of depression and that there were all these different factors that come in that were
Mm,
unexplainable as far as illness and so on. A lot of our immune system is tuned to our environment. And the immune system is highly dynamic with our behavior. So there's a connection that is so critical, which is one of the most fascinating things about the disease leprosy. I don't know if you have another 4 hours to talk about this, but leprosy is an epigenetic phenomena
Oh,
because the isolation that's talked about in Leviticus 14 when the leper is to set outside the camp. The isolation, just like with COVID, has a immunosuppressive factor because isolation can cause not only depression, but it literally gives a greater level of susceptibility to other diseases. So most people that
mm,
died of quote leprosy really died due to the trauma of isolation, and their genes were more susceptible to the cousin of Mycobacterium leprae, Mycobacterium tuberculosis, and most died from tuberculosis because of the isolation.
wow. That's crazy. That's
Isn't that
amazing.
fascinating? I mean,
mhm.
that's again, that's another epigenetic phenomena.
Hmm.
Yeah,
Wow. Let's talk about how this works for a pastor. So I'm a pastor in Baltimore City.
yeah,
There's
yeah,
a lot of difficult settings that occur in the lives of people who live in Baltimore City. Used to be when I moved here, it was
yeah,
the murder capital of the
uh,
country.
yeah.
Crystal leads the Relief Center and Baltimore
Mm,
Relief, and that's serving
Okay.
twice a week people that are without food.
mhm.
And so
Mhm.
I think we're both reading the book through the lens of the people that we serve and love and
Absolutely.
care about. And
Yeah,
so let's talk a little bit about that. What questions do you have?
yeah,
So there
yeah.
does seem to
Yeah.
be a focus in the book, particularly on addiction. And addiction is
Uh-huh.
something that we encounter it all day, every day, everywhere we go for work and in ministry. And I would love to know for you why that particular focus and what inspired that.
Because it's so closely aligned with the theology that's coming from Augustine. And
Okay.
that's when we sort of look through Confessions and you get like his who he was as a person. Okay, fair enough. I would love to say he's a saint, but it looked like a real strong sex addiction. And within his addictive behavior, it's very explicit about some of the challenges that he had as a guy. And it was something that he could not escape. He found himself failing time and time again. And however much he quote tried. And there's something about a regimented, and that's why I sort of look at, the idea about 2 Peter, 1, 5, and 6 about, the cycle of redemption, like how this redemption process happens, and it's sort of like beginning with a cognitive pattern and sort of learning what it means to have this knowledge and understanding self-control and leading to perseverance and then the godliness factor and all those kind of ways that we grow through a challenging, especially during the times of the impulse that you don't have the cognition for it. It's just something that you're being driven by. But what is that driving factor? And it's interesting, and this is in a subsequent book coming up, is understanding this other gene that relates to oxytocin. And oxytocin is the hormone related to attachment and belonging.
Mm,
And that sense of it, and we talk about community, but when you know you belong somewhere, there's a transformation that happens in that gene. That sense of
mm,
these are my people. this is where I can be. This is what happens with the pastor when you connect with the people and the people love being there because they belong there. And there's an upregulation of oxytocin and attachment that gives us a greater sense of, that belonging and exercising the gifts God's given us because it's a trust that develops. And being in a attachment centered, trust factored environment is how discipleship actually thrives. And why we as leaders, pastors, teachers, scientists, anything we have that's our gift is primarily to direct believers to a belongingness and attachment in terms of you do matter. Just
Hmm.
eye contact.
Yeah.
Just it can be just the sense of touch is so powerful from an attachment perspective. Why do you think, and we look at Leviticus 12, for example, that's a classic chapter of attachment that when a baby is born, it is attached to the mother for 3 months. And that attachment is the development of this gene to be epigenetically developed into a need that develops this confidence and certainty that happens with the brain-derived neurotrophic factor, which again, it goes back to how is that, what other genes are working in this sort of potentiation of learning and memory and that whole chain reaction of that is fascinating. There's a lot of research done on this attachment disorders and so on. It happens when childhood adversity, you heard of the ACE test and so on. This early childhood adversity from zero, zygote, all the way to 7 years old is where all the epigenetic patterns happen and where we develop that sense of attachment. And from there, we have this sort of like this imprinted matter that this is who we are kind of concept. And then a traumatic experience comes up and how well we adapt to that moment. And that sense of adaptability has everything to do with the function of this oxytocin gene.
Hmm.
So,
Well,
yeah.
so the discoveries. So what's the difference between um, doing just making lifestyle changes? Because so you've got the science, you have the studies that are done on
Yeah.
epigenetics, and they're not
Yeah.
studying Christians. They're just watching and observing people making lifestyle changes, and they're saying, okay, wow, so this had it caused the gene to express itself in a healthy way. But then you're coming along
Mhm.
and you're saying, wow, this really syncs up well with the Christian faith and what's the life of a believer. What is the difference between somebody taking what we're learning from epigenetics and being a Christian? And
Okay.
what's better about being a Christian? And why not just say, hey, just make these lifestyle changes?
Mm. Excellent. That's exactly, oh my gosh. Okay. And so, how is it in that sense of, we can look at it from a social program, and the social program is exactly what you're describing. Just do these changes and it will work. And this is the way your genes function, and you should just be happy and enjoy.
Right.
And this is why I went to the original sin concept. Why? Because I would love to say that we're perfect machines, and that's kind of the way it works.
Hmm.
And one size fits all, just stop smoking, you'll be fine. And, but then interestingly, and this is kind of like an interesting footnote that goes with that, is that there was a study done in rats, and they were bred to prefer cocaine. And the whole idea about these rats is that they needed cocaine
Did you fund
administered
that study?
daily.
I can't
Okay.
say.
This study it came out in the early 2000s. And the study basically describes it as the technician, they're measuring the dopamine in the animals, and they have this, canalization of the brain so they could actually look at the quantitation of how the dopamine in they sort of like this graph of dopamine concentration versus time, and the individual would walk out with this cocaine, put it in there, and the dopamine would spike. But then they did an interesting moment. They slowed the time factor down So they could actually look at the nanoseconds, milliseconds or whatever. And they saw that it wasn't the cocaine that they were addicted to in terms of the dopamine surge. The dopamine surge was at its maximum as they saw the technician bringing the cocaine, i.e., it was the anticipation of
Hmm.
the cocaine that elevates more so than the cocaine itself, which is a very telling statement about worship in terms of our anticipation of the presence of God, which makes a Christian experience different than a secular experience. Why? Because living with the sense that God is present, living with this sort of anticipatory prayer, living in that and just like, what will he say to me today? That concept of anticipation is what we propagate in our congregations so that there is this sense of we're not addicted to the stuff, we're addicted to the presence. And this is the whole idea about the presence of God. He creates anticipation in us. And this is another biological phenomenon that we need to sort of harness as believers and realize that there is a solution that may not be pharmaceutical. It may not be this, you know, regimented standard that this is what you do. You change your environment, boom, it changes. There's been this whole concept of, oh my goodness, look what happens with a believer that's genuinely walking as studying the word or whatever we do as believers and all of the sort of disciplines of a disciple, the transformation happens physically with this concept of anticipating what he will do because it's relational, much more so than obligation, which makes a massive difference.
Well, that's great. Yeah, that's helpful. And I think that that's what I was getting as I was reading through the book. Um,
Uh-huh.
I had another question, but
Go ahead.
um, we were talking last night as through one of the latter chapters in the book about this. You throw out the idea that maybe with in Genesis 3 that the DNA stayed the same, but maybe that the fall occurred in the epigenome. Is that, am I reading that right? Is that an
Yeah,
idea that you're
you
throwing
are.
out there?
Right, right. But you got to remember also that when sin entered in, death entered in. So
Right.
the idea about how our genome was affected was our longevity was changed. And there's a whole idea in biotechnology and all that, understanding what's called apoptosis, which is simply the study of programmed cell death. And it has a lot to do with this enzyme called telomerase, which literally every time a cell divides, a piece of the chromosome is cut, and it's part of the process of dying. So aging is related to this function of this telomerase, meaning that we're all susceptible genetically to die. So the epigenetic change, though, is how sin was integrated into us
Hmm.
because it doesn't change our genome. It changes literally not only behavior, but it also potentiates this concept of not only aging, but it also sort of turns on another pathway that allows us to experience this dying, you shall die, that's talked about in Genesis 2. When he says, if you eat of this tree, it's meaning dying, you're going to live a dying life. And that is how epigenetics and genetics connect because you have sin in us and it's a sin nature and it's going to have an effect on, yes, we're going to die, but it's also in a state of you're going to be in a falling away process as well. So you have that's where we get this idea about when God saves us, he positionally sets us free, perfect sanctification. And then at the same time, we're also being progressively sanctified, meaning that there is this ongoing epigenetic phenomena that's happening even in that dying process.
Hmm. Well, that's a wild.
Okay.
Um,
It's really important to look at that in Genesis 2. Yeah.
Yeah, man, that's so crazy. Um, the Can I ask another question?
Yeah, of course.
All right. the So when we talk about spirituality,
Yeah.
and then I read your book, one of the things that I often think of spirituality as very immaterial. But the more I'm reading your book, it sounds as if you're identifying our spirituality more physical than we may have realized.
Mhm.
Is that fair?
Yeah, very fair. Absolutely. Absolutely. You remember our genes, the way our genes produce proteins are tuned. And when the tuning changes, the types of proteins and or the frequency or quality is altered as well. So anything around that space is an epigenetic issue. So that to say is that what I do, how I exist, how I respond, the tone of someone's voice, the reactions, all that has an impact specifically on these types of genes that we see evidence of in behavior and or whether it be internally like immune function of the liver and kidney, all of those, interestingly, are changeable dynamics that can happen with environmental factors. So we're literally in this constant dynamic.
Hmm.
When we were born, we were not put in stone and saying, this is the way you're always going to be. Notice that we're all aging. Notice that we're all behaviors that we thought, oh, I used to be so weird. You look back at the behaviors that have been extinguished in your life and realize that just didn't happen. God allowed that to happen as we're being sanctified. So we actually have a diagnostic way of looking at our existence as Christians to see how we're becoming more like Him and decisions we're making and so on. I know we have a Romans 7 problem at times. We do blow it, but we're not habitually blowing it. And that's the big distinction. Habit is a fascinating concept in terms
Hmm.
of this concept in neuroscience called neuroplasticity. Have you heard that
Yeah.
term?
Yeah.
Neuroplasticity, like how we actually have the sense that neurons get strengthened in our when we put our minds and focus towards a particular behavior and habits form with by making this new trough in our neurons so that we're having greater levels of connection in the synapses within a given behavioral change. So when I literally quit smoking, I learned that the best way, instead of trying to stop smoking, was to focus on something good for my body. So I started, it literally had to be an internal conscious awareness that I'm hurting myself. And I know it's dumb sense, but it's a learned process. To start with basic stuff and habit changes that I had to agree that this is wrong. And in that agreement, I also know that what is right is doing something better for my body. I can't eat this, but I can eat this. So learning that decisive nature of what is good versus not good or evil, if you will, is that discernment factor that happens, yes, with God's word and by the Holy Spirit. He equips us to make these decisive, ongoing decisions, which is really why, again, it goes back to a Christian um, perspective is going to have a much higher capacity for lasting results than one that's secular. So we have to keep that in mind because by doing this just by the conditions that we're in right now is a very limited and can be often, sadly, the longevity is very limited as well.
Hmm.
So, you know, um, it always becomes a challenge at times because I want so bad to just treat Jesus like a pharmaceutical when, he's much more than he's a person. So that's where this attachment factor comes in. And the concept of he loves me, what does that look like? How do I know I belong to him and to someone else? We just know. And
Hmm.
how is this concept of we just know? Is the power of the Holy Spirit that overrides our questioning whether that happens because we genuinely let it happen. And that's kind of what happens with, you know, a spiritual nature that God's given us.
Hmm.
So,
Man, I would love to
you
be in your
know?
head, Lou. You got so many different things going on. It's amazing. You sound a little bit tortured in that, like, you're so excited and you are understanding all this science and you love Jesus and you're like, wow, these things
Yeah,
connect. And,
they do.
there is this
Yeah.
complexity to it as well. So,
Yeah.
yeah,
Yes. Yes.
yeah.
Yes, I agree. And that's one of the challenges, but it's also if you can get it semi-digestible and sort of see and integrate it into some of the thinking in terms of pastoral care, in terms of recognizing this person is in this condition, but there is hope because there is changeability, because we can access that gene of oxytocin simply by being there. It's amazing what the ministry of presence does.
Hmm.
It's miraculous. And it's how God uses us in ministry. It's not, we don't have all the answers. We're just part of the process of getting them to the answer. So that's why we got to guard against rock star pastors. They're meant to be conduits. They're not supposed to be the final answer. The people have to be empowered with these tools, which is one of the reasons why, yes, I would love to elaborate more on the tools that in this book, but I got to get the paradigm right first for them to get the understanding of it. And gosh, that's really the essence of it.
Well,
So taking Romans 5:12 as it is, that we're in him, that the sin of Adam is in the human race. It's spread to all of us. And the only way we can, that I've seen, and that is even explainable, is an epigenetic process. And I end the book with some kind of idea, although we don't have the absolute nature of that, it may be something that's quantum. And we just, as they almost put that almost as a prophetic statement at the end of the book, because, talking with mathematicians, they're saying this is a process that could actually happen. And I think that's something we just need to be mindful of the fact that, when Jesus is in me, he's doing something in me. He's got all of this. He holds me together. I mean, if you look, Colossians is so profound in that regard
Hmm.
that understanding that he's holding every one of our molecules in that level of sovereignty sort of supersedes all the names we want to call these molecules. The idea is that he's doing something, and then the more I learn about it, the higher my responsibility is to know that I do know. And I know that if I go there, if I do this, if I eat this, I know that it's going to be a problem, not only spiritually and physically and emotionally, because there's evidence in all those categories.
Hmm.
So,
Wow. Yeah, I think
yeah,
it seemed like one of the undercurrent themes was just the idea of hope
yeah,
and
yeah,
that you're not a victim. I mean, so in Baltimore City, there's some dire circumstances that kids are born into, and
yeah,
one of the things that you're testifying to through science is just saying no. The way God's created us is there's great hope. Um,
yeah.
yeah,
Yeah,
yeah.
exactly, exactly. And as in your ministry, God love you, bro, is that, the two of you are in these really interesting spaces of looking to see, yes, bringing it, but what is going to generate this sort of like this functionality of these, particularly these genes we're talking about, to give them a greater sense of of learning. And once the belonging starts, is when the learning starts. These work together. They're two different yet highly connected concepts. Of once I belong is when I can start to deepen my learning
Hmm.
and I can understand. And my understanding is affected. And I validate. God's using that to sort of allow transformation to occur. We're giving the people the space to become who they really are in Him.
Hmm.
And, that's kind of what our mission is on planet Earth, is to bring about that. And it's God's word that is transformational
Hmm.
and just letting it be what it is. And the clearer and the more equipping we are with it, oh my gosh, it's amazing what it does to people's lives. It's just, it is just
Yeah.
miraculous. I've got a very good friend who's also an epigenetics professor also, and he's Jewish. And, you can get shunned so easily because, the Jewish mindset so often can be, that there's a lot of legalist factors and so on and keeping all that going. And it's not like that I'm not dismissing it. I'm just limiting it to the fact that we're dealing with grace here. And God's grace is the most potent, overwhelming sense of that oxytocin. And, giving a sense that, yes, I blew it, but I know I'm forgiven. I don't have to do anything out of guilt, really, because once he's forgiven me, that is probably one of the most powerful epigenetic phenomena there is.
Hmm.
That forgiveness
Yeah.
goes biologically. It isn't just a spiritual thing.
Hmm.
It does transform us to not, and forgiveness is the wide open door to belonging. It is exactly what it is. Because when you forgive somebody, you connect with them because that's how I love you. Is I not only forgiven you and or like whether the wrong is relate it doesn't matter. There's something that happens within these genes that does the concept of attachment. So attachment disorders is something that, it'd be really interesting sometime to look and see what happens when in different families and make an assessment of how their early life adversity, what kind of life they were grown in, and what are some of the changes you can bring to sort of ameliorate that sense of, you know, you were abused or you were, disliked or you're nutrition, all that stuff. Seeing that and looking at it from a merciful perspective and seeing how these genes can be altered, you can do your own epigenetic studies right in front of you and see the transformation.
Hmm.
That's what makes it so powerful. These are
Yeah.
not just anecdotal ideas. We've seen them, especially with PTSD. Oh my gosh, that is like the classic as of all. So
Well, well, Lou,
yeah,
thank you. We could keep going.
yeah.
There's a lot of ground that we could cover. Um,
Uh-huh.
Anything final that you want to share, Crystal? Final words.
I think just thank you for giving hope, um, for bringing together the science and the theology. That's hopeful, I think, for our community, for our society. But then I know a lot of folks myself who don't give as much credence to the theology. And so what you've done in so many ways, just it points to God through the science. And that's remarkable and
Mhm.
so powerful.
Mm,
And so I thank you for that.
yeah.
Yeah.
Yeah, Lou, thank you. We appreciate it. Hopefully, we'll be able to do this again in the future.
Absolutely.
Yes.
Absolutely. Yeah.